So much drama in the LBC. It's kinda hard being Snoop D-O double G. But I somehow, some way, keep coming up with funky ass shit like every single day.
Hi guys, my name is Nako Nakatsuka. I'm a 4th year chemistry PhD student at UCLA. And today I'll be helping you guys out by going over some general chemistry concepts. And good luck with the course. So in 1939, they found that DNA is a polymer of many nucleosides linked by a 3' or 5' phosphodieesters. And so in this case, let's say this is the 5' end, and that this is the 3' end. And basically all the nucleosides that compose the sugar and the base was linked down like this with the phosphate backbone. And so the nucleotide sequence gave the DNA or RNA primary structure. And so primary structure of DNA or RNA was the nucleotide sequence. Just like the primary structure of proteins is the amino acid sequence. Now we just have different building blocks. And in 1957, the Nobel Prize in Chemistry was awarded to Lord Alexander Todd for his work on nucleotides and nucleotide coenzymes. But before 1944, which is kind of crazy, proteins were believed to carry genetic information, and DNA was just a mere biological curiosity. But finally, in 1944, DNA demonstrated to be the cellular molecule that carries genetic information. And now this is common knowledge, right? And in the 1950s, Erwin Chargaff studied heterocyclic base ratios in DNA from various different organisms. And in the DNA of all organisms, they found that the A/T ratio was approximately 1:1, and then that the G and C ratios were also approximately 1:1, and also that the G plus A over C plus T ratio of all the bases, which is basically the purines over the pyrimidines, also gave you a ratio of approximately 1:1. So this is called Chargaff's rules. This was in the 1950s. So this was another very important thing because in DNA you have these 4 bases, right? Right here we have the adenosine, the guanine, the cytosine, and the thymine. And by Chargaff's rule, you can figure out that A is probably bonded to T because they're in a 1:1 ratio, and G probably interacts with C since they're in a 1:1 ratio. And another thing that they found was that the base ratios are random in RNA. So in 1953, Rosalind Franklin performed X-ray studies of DNA to show a helical structure. And so this is an actual XRD result of looking at the DNA structure. And he found that the diameter of these helical structures was 20 angstroms, and that the length was 34 angstroms per 360 degree turn, and he was also able to calculate the density. And so just from XRD results, he was able to get all of these parameters for a DNA helical structures. And then from here, James Watson and Francis Crick combined Franklin's XRD data as well as Chargaff's rules with the ratios of the bases, and they also examined molecular models. And from here, they came up with something that would give them the Nobel Prize in the future, which is coming up with the structure of DNA as base paired in a double helix. And this double helix structure is actually the secondary structure of DNA. So remember, the primary structure is the arrangement of the bases, and the secondary structure is the double helix. And so let's look at these Watson-Crick base pairs. And so we have the adenine and thymine bonded together via hydrogen bonds, and we also have cytosine and guanine bonded via 3 hydrogen bonds. So adenine to thymine was found to have a double bond, whereas cytosine to guanine was found to have 3 hydrogen bonds. And the base pairs are similar in size and shape, which allows their efficient packing into a double helix. So this was a really momentous discovery of the double helix for the DNA structure. When we look at the base pair double helix, it has major grooves and minor grooves, and these dimensions were determined with the X-ray crystallography. And so the major and minor grooves result from the overall geometry of the Watson-Crick base pair. And so if you look at an AT, TA, GC, or CG base pair, you'll notice that the glycosidic bonds are in about the same place in all the base pairs, and that they are oblique with respect to one another. And so if you look at these diagrams, you can see that. And so this figure basically depicts an AT and GC base pair, and if you extend the glycosidic bond in each base pair to form an imaginary line, the lines will form an obtuse and an acute angle. The major and minor grooves form between the phosphate backbones of the DNA molecule. So the obtuse angle forms the major groove, and the acute angles form the minor grooves. And so that's basically it. If you look at these diagrams, this is the major groove side, this is the minor groove side, and this is the major and this is the minor when it gets structured into this double helix. So the last thing I wanted to talk about was the DNA space problem. And so the human genome consists of about 3 times 10 to the power of 9 base pairs. And so by using these parameters, if I did 3 times 10 to the power of 9 base pairs multiplied by 34 angstroms per 10 base pair, multiplied by 10 to the power of negative 10 meters per angstrom, you would approximately get 1 meter in length in order to fit in all of the base pairs in your body. And so since that sounds a little crazy, the solution that biology has created is that the DNA tertiary structure is a supercoil. And so DNA is found in these supercoiled tertiary structures so that it can store so much more genetic information and not be 1 meter in length in your body. And so you're basically squishing in so much information inside your body, which is representative of DNA. So I hope this organic chemistry review was really useful to you guys. Good luck in the course and just keep studying, and I'm sure you guys will do great. With my mind on my